Welcome to our exploration of the Ascidian larval form. If you have ever wondered how simple, stationary sea creatures could possibly be related to complex vertebrates like humans, the answer lies in this microscopic, free-swimming tadpole. Today, we are going to uncover its fascinating anatomy, its biomechanics, and the bizarre transformation it undergoes. Let us dive in. To begin our journey, let us look at a fascinating analogy to truly grasp the life cycle of a sea squirt. Imagine a highly advanced spaceship landing on a planet, gluing itself to the ground, and transforming into a simple water pump. This science fiction scenario is exactly what happens in nature. The Ascidian larva is our spaceship. It is a highly complex, free-swimming creature equipped with a sophisticated nervous system and a strong tail. It actively navigates the ocean, searching for the perfect rock. Once it finds that rock, it undergoes a drastic transformation, losing its brain and mobility to become a stationary adult. Now that we have a conceptual picture of this creature, let us define it biologically. The Ascidian tadpole is the embryonic stage of marine animals in the subphylum Chordata. What makes this larva biologically spectacular is that it perfectly exhibits all four fundamental characteristics of the phylum Chordata, traits the adult completely lacks. It has a stiff notochord in its tail for skeletal support and a dorsal hollow nerve cord that forms a sensory vesicle with organs for balance and light detection. It also features pharyngeal gill slits for respiration and a muscular post-anal tail for propulsion. Having discussed these biological features, let us see exactly where they are located. On screen is a cross-sectional diagram of the Ascidian larva. At the very front, you can see the adhesive papillae, which it will eventually use to attach to a rock. Just behind that, in the trunk, is the sensory vesicle, containing the ocellus for detecting light and the statocyst for balance. Notice the dorsal hollow nerve cord running along the top, and right below it, the stiff notochord extending through the tail, surrounded by muscle bands. This specific arrangement is what allows the larva to swim effectively. Seeing this complex anatomy brings us to a crucial realization about animal evolution. Before scientists studied this larval stage, adult sea squirts were mistakenly classified alongside simple invertebrates like sponges, simply because they looked like stationary blobs. However, the undeniable presence of the notochord and dorsal nerve cord in the larva proved beyond a doubt that these creatures are actually chordates. This makes them close evolutionary cousins to vertebrates, including humans. Without this crucial larval stage, the evolutionary bridge connecting simple marine invertebrates to complex backboned animals would remain a confusing mystery. Understanding their evolutionary importance naturally leads us to ask how this remarkable anatomy functions in motion. The larva relies on undulatory swimming, meaning it creates wave-like motions that travel down its tail to push against the water and generate forward thrust. This mechanical action is entirely dependent on the stiff notochord. When the tail muscles contract, the notochord prevents the tail from simply telescoping or collapsing inward. Instead, it forces the tail to bend from side to side. This brilliant biomechanical design translates muscular energy directly into forward kinetic energy, allowing the larva to navigate the ocean. To quantify this biomechanical movement, we can model the larva's swimming efficiency using a fundamental kinematic equation. The formula is V equals K times F times A. Here, V represents the forward swimming velocity of the larva, measured in millimeters per second. The symbol K stands for the efficiency constant, or slip factor, which accounts for the friction and viscosity of the water. The variable F is the tail beat frequency, meaning the number of side-to-side sweeps per second, measured in hertz. Finally, A represents the amplitude, or the maximum distance the tail bends from the center line. If the larva increases its frequency or amplitude, its velocity increases proportionally. Now that we have our formula, let us see this with a concrete example. Imagine a marine biologist observing a larva actively searching for a settlement site. We are given a tailbeat frequency of 10 hertz, an amplitude of 0.5 millimeters, and a water efficiency constant of 0.8. We need to find the forward velocity. We start by substituting our known values into the equation. First, we multiply the frequency of 10 by the amplitude of 0.5, giving us a theoretical maximum speed of 5.0. Finally, we multiply by the efficiency constant of 0.8 to account for water slippage. The final answer is 4.0 millimeters per second, a perfectly adequate cruising speed for finding a home. Having successfully calculated the baseline velocity, we must now consider what happens in a life-threatening scenario. If a predator approaches, the larva must reach a specific target escape velocity. To find out how hard the larva must work to achieve this, we need to rearrange our original kinematic equation to solve for the frequency, F. We start with our standard formula, V equals K times F times A. Our goal is to isolate F on one side. The first mathematical step is to divide both sides of the equation by the efficiency constant K. This begins the process of working backward from a desired physical outcome to find the biological effort required. Continuing from our previous step, we now have the velocity divided by K equals F times A. To completely isolate the frequency, we must now divide both sides by the amplitude, A. This moves all the known environmental and physical constraints to the left side of the equation, leaving the biological variable, F, completely alone on the right. We then simply flip the equation around for standard readability, giving us our final rearranged formula. F equals V divided by the product of K and A. We have now mathematically proven how to determine the exact tailbeat frequency required for any target speed. With our newly derived formula in hand, let us apply it to the predator escape scenario. The larva detects a shadow and must immediately accelerate to a target velocity of 12.0 millimeters per second. The efficiency constant remains 0.8 and the amplitude is fixed at 0.5. We substitute these values into our rearranged equation. First, we multiply the denominator values, 0.8 and 0.5, which gives us 0.4. This represents the effective forward distance gained per beat. Dividing the target velocity of 12.0 by 0.4 gives us our final answer. The larva must violently whip its tail 30 times per second to escape, a massive spike in muscular exertion. After surviving predators and successfully navigating the ocean currents, the larva finally reaches its destination. Once it finds a suitable location, it attaches itself head first using its adhesive papillae. This triggers a bizarre biological process known as retrogressive metamorphosis. Unlike most animals that grow more complex as they age, the Ascidian actually degrades. It completely absorbs its beautiful muscular tail and loses its advanced nervous system, including the sensory vesicle. It transforms into a simple, stationary filter feeder. It literally sacrifices its high-level biological complexity in exchange for a life of permanent stability on the ocean floor. To wrap up our lecture, let us review the key takeaways. The Ascidian larval form is a biologically monumental stage that proves the chordate lineage of sea squirts. By proudly displaying a notochord, dorsal nerve cord, and postanal tail, it serves as the evolutionary bridge to vertebrates. We also learned how its stiff notochord enables efficient undulatory swimming, which we can calculate using kinematic physics. Finally, we saw how it undergoes retrogressive metamorphosis, shedding its complexity to become a stationary adult. You now have a comprehensive understanding of this incredible biological marvel.